Yeah, I think good morning. You know me, right? Is it okay? I can stand like this. So the topic is the role of photosynthetic pigments in chloroplast biogenesis. So we'll be discussing about the pigments. What are the pigments and their importance also we'll discuss. How they are placed in the photosynthetic membrane that we'll discuss uh, maybe another 40-45 minutes. So we all know that. I mean, the Earth, when I mean, 4 billions years ago, there was no oxygen. I mean, there was no gases. You see the left panel where you have the Earth was like this. So then started with the air. You have the burning kind of thing. Then there was the gases has formed. Then what you see here at the moment right now, the green phase, whatever this one, all these 1/3 of the water, you have, and also the green belt, and the photosynthesis is evolved such a way that the plant, we are all surviving because of the plants. So now you all know that why I kept this slide. Maybe you can also get anywhere in the internet. So we all like sun, right? So if you don't have sun, and you will be frustrated, and no, you may not be feel fresh. So that's what, when you do, most of the people, you do Surya Namaskar, right? Those are, this is somewhere in a river, somewhere. This is like, to get something, a juvenile, I don't know, or thing, you can say that you get something freshness and you start the day with that. And the birds, they fly, if there's, sun comes, the birds fly, all animals will be very happy. And, anyway, this from sun, you are getting energy. You say that positive energy. The mythologically, or it's true that you get a positive energy. I'll tell you a story. Like most of the European countries, they get almost 6 months they don't have sun. You know that, right? Especially Norway, Sweden, Russian, some part of Russia, many other in France. What they do, what they do? If they don't have sun for 3-4 months, you will get not frustration. It's a kind of I'm not getting the word. Huh? That is the thing. But you may not be like feeling like something like
Laziness.
energy, whatever. So laziness, whatever. What they do is they go for phototherapy. How you go for a gym and all. They do phototherapy, means light. The rooms will be full of light, which is mimicking to the sun, and they stay there just after the hours, after the working hours, they go and stay in the sun, like artificial sun. If they don't get whatever vitamins, whatever you think, whatever you think, so it will be a deficient, so they may have other complication with the one. So likewise, I said, all this sun is very important. Now what we are also now producing a lot of solar energy, and many more things we'll discuss. So now the basic um, photosynthesis, it started with anaxidenic. After this, the archaea, say that archaea bacteria, they started with anaxidenic photosynthesis, where they don't fix CO2, but they use organic stuff like H2S and gases, and produce the ATP through purple bacteria, non-sulfur bacteria, sulfur bacteria. These are the bacteria. Then after that, about 2.5 billion years ago, the first oxygen species, cyanobacteria, evolved. Then you could see all these one, the autotrophs. That means autotroph is nothing that can synthesize their own food. And so then, the basic reaction of photosynthesis, I mean, you all know that. I don't need to explain. So it requires solar, that means light energy. and whatever the light energy will chemical energy that will need, you have a two cycles. I mean, light dependent, light independent. Light independent is also called dark cycle or Kelvin-Bunsen cycle. The whatever ATP is produced, ATP and NADPH is produced from linear electron transport, that will be utilized in Kelvin-Bunsen cycle to produce the all biomolecules, essential biomolecules like carbohydrates. Then it goes to all lipids, proteins, and all be synthesized. That's what you could see here, the carbohydrates, and of course the water molecule and oxygen. So now, why plants absorb only in the visible region? So this is the electromagnetic spectrum. You have a gamma rays, X-rays, UV rays, and the region is called visible region, 400 to 700 nanometers. Then infrared waves and radio waves. These are the the electromagnetic spectrum where plants absorb only the visible region. So can any of you tell why plants absorb only visible region? Of course, they absorb in UV region also, but the UV region maybe damages the plants. So plants can the photosystem, not only plant, photosystems will be get, and they absorb. They have a photosynthetic, the receptors where they can take it, they can absorb. But however, they may be deleterious to the plant. Of course, other radiations also. I mean, we don't get other radiations, and so in electromagnetic spectrum mostly we get near UV and little bit infrared. So where plants can absorb, but they have a mechanism to they cope up or a protective mechanism. So why plants are green? So it takes Websier color. What we say that different wavelengths of visible light are seen by human eye as a different colors, but they absorb. But they can be also plants are green, but they are not green, right? The eye which we are seeing, because of reflection, you could see because whatever the light it takes, it takes red light. How we are seeing it blue, I mean green color. So that is all because of the reflection. So this I don't want to discuss because it has been discussing many people. Yesterday also, Das sir was discussing, but these are the chloroplast. So now we'll talk about these pigments. The topic it says pigments. So plants have evolved with the various kind of pigments. The pigments are these are called the major pigment is chlorophyll E. The all others are accessory pigments. That means they help transferring the energy from these to chlorophyll. So now I showed you this picture. So looks so beautiful, right? So now the green plants, are there the plants, the pigments or the chlorophyll pigments will convert into the carotenoids. The carotenoids have many other pigments. I will discuss bit later. I mean, we can tell later. So carotenoids have xanthophylls, zeaxanthin, anthocyanin, luteins, like several pigments. They have a different colors in plant depending on the plant. Based on the plant, they change the colors in different colors. So these now, I mean, if you those who are going, if you are somebody visit European countries or Canada or in US, it's a big tourist place now. Right now, I think it is the September, October. So people go and just to see this beauty of this nature. I mean, night so first it's green, then it convert into the yellow color. From that yellow, it convert into reddish, pinkish colors, various kind of colors. It changes because carotenoids convert into, once it is chlorophyll convert into carotenoid. Carotenoids will be then carotenoid, many carotenoids, like xanthophyll, zeaxanthin, anthocyanin, lutein. It's a phase wise. The conversion will be 1 to 1. It takes days, few days. Then you see that the colors. So how we can measure? We measure the chlorophylls? Yes, just take organic solvents, acetone or anything, methanol, DMSO, they can dissolve. You just crush it, the leaves, or even the cells, cyanobacteria, they just grind it or mix it, mixture, blend anywhere. So you can get all these pigments. So then you measure a simple absorption spectra where no, they have wavelengths, different wavelengths, and you can characterize whether the chlorophyll. I mean, it's already standard, standards are there, or you can also the fixed wavelength is already been in the literature. From that you can identify all these peaks. Let's say this one, this is the chlorophyll A peak. Let's say this is the chlorophyll B, and this is the carotenoid peak. So likewise, you can understand the nature of nature means how quantity you can quantify it. So then the structure wise, these are the chlorophyll structure. A simple change in the groups of this, you can see here. It's simple. I mean, these are the phytol ring, the heterocyclic compounds, and majorly magnesium is the one, determines the green, the nature. Will same one in human. What is that? It's same one, same structure. It will be iron will be there. So hemoglobin. So we will have a iron. So then it will be the same, almost same structure. So the simple change in this, the methyl group to aldehyde group, it changes the group. The wavelength, absorption capacity also, like 663 is a chlorophyll A, then it goes to chlorophyll B is 652. So there will be about almost 12 nanometers. The shift will be there. So now they almost beta carotene, of course, this aromatic ring, and zeaxanthin. Also, you see that zeaxanthin same ring. You have a additional OH group. And this will tell about the zeaxanthin structures. Of course, this chlorophyll C, there are plants, most of the land plants have chlorophyll A, B, chlorophyll A, B. But there are bryophytas or lower organisms. They will have, those who stay in those who live in extreme conditions like, salt or, high temperature, high altitude. They will have a different, the same carotenoid, chlorophyll pigments, and even different carotenoid pigments also, up to F, chlorophyll A, B, C, D, F, now pigments, and even now they identified G also, G. So, but it is only the little structural change, but they all looks green. And can we measure all of them? This is, you can see here, all the chlorophylls can be measured. Again, as use acetone, methanol, just simple. You can see the structural changes, a small change in the structure, chlorophyll A, B, C, D, E. I said, I can go with many these are the bacterial chlorophylls, and these are the land plants. What you have, chlorophyll A, chlorophyll B. These are also same structures. The only the phytol ring, there is a functional group. One functional group change can make lot of differences. Like bacterial chlorophyll, they absorb in longer wavelength. That is about around 800 nanometer, 900, even 1100. Where our Dr. Yusuf will be talking about the topic on Saturday, right? Day after tomorrow. So he will describe a little bit more on how they can, So you can isolate all these one. Now, here, the additional, there are two, the pigments, bacterial chlorophylls, and you see that it absorbs somewhere 800 nanometer. There is phycocyanin, allophycocyanin, phycoerythrin, where the cyanobacteria, they have these pigments. So we'll discuss about all those one later. Okay, so about these are the pigments. And I've been, Chiro Takashi and other people, Rebidas were talking about more about structure and the composition, biogenesis. So what are the application? But now many of you know that application, but through photosynthesis, what kind of application we can generate. So you people can, looking at this one, maybe you may have your own ideas, and your perception will change now. So this we know that, anything, whatever we are eating, this is only because of plants. Plants are, whatever. Because it's a cycle, life cycle. The animal system also has to eat, the plants, but that is again, through photosynthesis only, all this will you get it. So I mean, I kept this one to pollution controls all the plants can control pollution. What is this one? This picture? Oh, fog in Delhi. But what is that one inside? India Gate. So maybe in Australia you have seen that like that, right? That color. So this is more of fog with pollution, smoky type weather. But if you have more plants, they will try to, can reduce this one. So now the pigments, chlorophyll, carotenoid, anthocyanin, they looks like this. So now what are the importance with the pigments? I mean, this also, maybe they all know that I teach this one in the class also. So now I mentioned here phycobilisomes. It's not only phycobilisomes, any pigments, chlorophyll, anthocyanin, lutein, all of them have this kind of roles. So we can use this one in pharmaceutical industry, and they will be acting as anti-inflammatory, neuroprotective, hepatoprotective, hypocholesteric. All this will be helping this one. You know that spirulina platensis, what is this? It's not algae, it is a cyanobacteria, blue-green algae. So, and chlorella, and there are many other, the cyanobacteria or algal system. Chlorella is algae, green algae. So people take it as a protein rich, and it also protect. It's proved already. The research has been, already, a lot of research has been, carried out in many places where, all these functions they have. So for girls, most of your girls here, so all these one colors, I mean, lipstick, sun protecting eyeshadow palettes, all can be used by using these pigments, because the cyanobacteria phycobilisomes, they have three kind of pigments. These are the pigments, phycobili proteins. So it's like, this color, and bluish color, a light bluish color. These three colors, independently you can make whatever the colors you would like to have, and these are all highly anti-inflammatory and anti-diabetic also. So, and also you say that therapeutics, Alzheimer's, aging, yeah, I think, I just missed with HRO, if you have been there. So my students, I think most of my students visited Japan. So every meal they wrap with the cyanobacteria. Actually, the red algae are cyanobacteria. Sushi, Japanese food, sushi. What are other foods? Onigi, whatever you say that. With rice, they wrap with that. Any food, or they also use the seaweed, which has a highly abundance of the pigments, high pigments. It's all because of one. So now HRO and his wife, if you see them, how old they are, you can predict how old they will be. 40 or younger than me. So anyway, so Japanese, Koreans, they live longer. Maybe one of the reasons, every meal it comprises with the wrap with this, the cyanobacterial or red algae, which contains high amount of phycobilisomes. So you want to live longer, you can use these pigments. Okay, we discussed antioxidants, and they'll be very interesting. This one, I mean, some of, not all proteins are, they give fluorescence. So what we do is, you can take this one as a probe, as a phycocyanin, phycoerythrin, or allophycocyanin. You can use this one and for a tag this one and get the colors. So we also, that can be also used. And more than that, we use dessert, milk product, ice creams, now it also it been using for all this, the food stuff also. So this is again same thing. Bacterial chlorophylls also will have a same kind of pigments. I don't want to talk about more on this one. Only thing, even colors, textile color also will be used. A lot of ecological applications that we'll discuss. Okay. And as I said, specifically pigments and carotenoids, what it does, the same kind of functions. It has a more of, the plant, if you take nowadays, I will discuss that one later. Astaxanthin. Have you heard astaxanthin? Astaxanthin is again a carotenoid pigment, where people are now working a lot on these pigments to understand. I mean, not understand, for industrial applications, not industrial, therapeutic application, but industrially they develop and they grow any cyanobacteria or algal samples and improve the astaxanthin. 10 mg is about 40,000 rupees, very expensive. Maybe we'll have one day tour Bionest upstairs, all of you. You can see that we'll have an industrial tour one day. This Saturday is packed, right? This Saturday is packed. We'll see that. Maybe this Saturday or 1 hour, we'll go over there and you can understand much better. Okay, again, same kind of a lot of importance. Have more carotenoids, and with the whatever the plate, you will have more of, you live longer. Okay, so now all this happens because of the high yield of photosynthesis. So now, what are other importance with the algae? Of course, in the plant also, you can convert in anything. If it is a biomass of plant or biomass of microalgae, you can get all these applications. So now, photosynthesis is not only give food, it gives many other elements. You see that valuable products, biofuel production. Valuable products means a small molecules where minerals, fertilizers, lipid fractions, lipids, lipid fraction will be used for many other things. So, you can, lipids also can be, whatever, the oils, you can convert oils or, what we eat, a lot of the oil stuff, antioxidant pigments, and there all be, what are the uses? These are the uses. Antioxidant pigments, vitamins, protein fractions, healthy supplement, and can be biogas, and carbohydrate fractions, health supplement, and they can also carbohydrates. The more the carbohydrates, especially plant, plant biomass, same thing, you put it as a plant, the biomass, you can get a nice, bioethanol production can be done. If you have wholesale biomass, they can make a health supplement. For example, I said wholesale means like, spirulina, a chlorella, even, the some of them like, other pigments like a dunella, where Pavitra is here. Oh, okay. So dunella, or you can also make the tablets of the hematococcus. Several, there are several algal systems. You can make health supplements. So then microalgae biorefinery is a very important nowadays. Is beyond photosynthesis. So people, to increase biomass, you need to have more photosynthetic performing algal system or plants, where once we have this one, so what do you do? Biochemical process. What if you do this process, take these microalgae and do the processing? Then processing is the one where you get fermentation, anaerobic and photobiological hydrogen production. Using that one, you get bioethanol, biogas, biohydrogen. So all these one are energy compounds. Bioethanol can be used as a fuel. Biogas, you can use fuel for cooking. Biohydrogen now alternative fuels. It's called alternative fuels. You can replace the petrol, where you can biohydrogen can be used. The lot of research is going on to improve the the biogas, bioethanol, or biohydrogen, because the resources, natural resource of petrol, is going down. So the alternative source is only the biogas and a biohydrogen. So if you go with the, these are the process. What are the process? If you go with the biochemical process, you get these products. If you get thermochemical process, more of heating energy. So that is a pyrolysis, gasification, liquefaction, compunction. So using that one, you can get all these gases, mostly gases. So trans-acidification, more of the lipids. If you have more lipids, so what you can get, you can get this one biodiesel, this trans-acidification. Mostly now, if you have more of fat, so where Jerome is here, Jerome Xavier, where is here? So what he is also doing on that same topic, to put more of lipids using CRISPR technology, and you get more of these trans-acidification, then improve these biodiesel products. So these are the some of the very important one. Based on this, a lot of people are now started their own companies, startups, and getting there are success stories, especially the we'll discuss about that a little bit later. Okay, this is the same. I don't want to go into this one. So this is one of the algae, that is a Hematococcus. Hematococcus is a green algae which produces high amount of the astaxanthin. I think we may have in the lab. Maybe you can see later. I think our Vijay is there. Vijay can show. Vijay Bunda, do we have Hematococcus plants? It's a reddish. Okay, so maybe he can show you in the during practicals time. So it will be like this, and it will be like this, and it will be like this. So I will show you one video where I'm advisor and also a kind of don't say a partner, but I'm advisor there. This is our after my advice. Now they got all this very nice one. So initially we grow like this. The cells, it's a Hematococcus cells. It's a green algae. So, and once you grow green, what we do, we transfer to the ponds, mini ponds. Here, mini ponds. So, and then it goes to the bigger ponds. Now we'll see even much bigger. It will become high yield of photosynthesis. The photosynthesis will get yield, and when it get saturation of these supplements, the food supplement or a nutrient, then they will be convert into chlorophyll, convert into carotenoids, especially the astaxanthin. So this was Cherish, right? Yeah, Cherish, where we collectively have done this. It was in Mitchell. It's an open pond system. So it's not only this Hematococcus, even Duinella. I think people are doing in Duinella, many other the algal system. So where okay, I will show you this life cycle. There will be so they have flagella. I mean, looks green, greener. So they're moving nicely, almost converting to the red color. They'll be still alive. Even they contain more of carotenoids, they'll be still alive, the cells. So it happens cell division during the nutrients deficiency, then they will become green again. When nutrient is there, they will become green. You just take them again, put it in the fresh media, they will become very green. This is a cell death, they can also happen cell death. For all these, photosynthesis is very important. Even it is converting carotenoids means there is a photosynthetic pigments play major role.
In the mature stage, when they have a lot of carotenoids, at that time, they go for photosynthesis or the external carbon source?
No, they do photosynthesis only. No, the nutrient you are talking, when it's getting nutrient is less because less carbon is there. What it does, to protect themselves, cell and want to protect. So there is it's a kind of a senescence. It's leading to the senescence. So the senescence is the end of the, you say that end. But it is the beauty of this one. It can, when you give again, the cell division will be slow. Take them and put it in the green again. The regular nutrient, they will pop up immediately. Cell division happens. They will be one. But when the nutrient is there is no CO2. There is no the ponds. Yes, they can take CO2. Even if CO2 is there, they have a cell senescence nutrient. Other nutrients will be less. It's not only CO2. Many other phosphate. It will become evaporated, right? So then they can convert into the pigments. It's clear? Okay, so this is again Hematococcus, and the Twinella salina is one where our Pavitra is working, but I don't know whether Pavitra has seen any time this color in your culture, right? Even in the end also. The cells have enough CO2. They are nutrient. That's why. But this also one of the company in the upstairs they are working, but now they moved to some other bigger space. It's also having beta carotene, high content of beta carotene, and especially the Hyderabad is very good environment to grow this Dunella because of it required dry culture, dry environment, and especially this Telangana region and the Rayalaseema region. You can grow nicely this one. Maybe Rajasthan, will be much better. So Israel is the a leader for this, because they have dry weather. They're growing a lot on this. These are all the ponds. I have visited these ponds in Israel. The same pond which I am showing here. Okay. So, again, the same thing which I am showing you again and again. All these one, you can use protein source, lipid source, carbohydrate source, vitamins, carotenoids, antioxidants. That's why I said those are Koreans and the Japanese, they live longer because of this. The pigmentation. They use pigmented food. So, and I wanted to tell you this one. Other importance with the using chloroplast. Chloroplast genome is size is much bigger. You can do a lot of therapeutic applications. You can, for example, anthracene vaccine, several other therapeutic you can express and then purify it and can draw. The people are using chlorella, chlamydomonas, many other algal systems. Chlorella, and chlamydomonas a big chloroplast where they're using this kind of, you see in the cholera vaccines, any antigens, several things can be used chloroplast. That is another, apart from photosynthesis, they also use as well. we are also working on this topic. So where Yusuf is guiding one of the master students, trying to understand. So pigments have chlorophyll pigments and bacterial chlorophyll pigments, especially the carotenoid pigments. So when they absorb light, they emit, right? It has to emit. The mechanism here, I mean, this is the nanoparticles type. So if it is, let's say, a cancerous cell, and then you go with your, the pigments tagged, or pigments can be tagged, or pigments can be supplemented. So you irradiate with the light. So what it does when it is irradiated, this produces oxygens, oxygen radicals, like singlet toxins, mostly singlet toxins, or you can also produce ROS, where the cancerous cells can be, this LED light, or you can also use, if you are using a bacterial chlorophyll absorb light in longer wavelength, and when they absorb light in longer wavelength, infrared regions, infrared is more efficient to kill the cells. So for you can say that the photosynthetic pigments are given nowadays through for the radiation, mostly radiation, and even chemo also will be now doing with this one, and so that will be a lot of the cell death can happen. It is happening actually. There are a lot of research, and especially Israel scientists, they have invented this one. I thought they will get Nobel Prize, but somehow it is getting delayed for them. Wisman University, who invented, see that these are singlet toxins. It produces singlet toxins, and singlet toxin is again this radical. It can go and kill the cells, cancerous cells. Of course, there will be that's why what they do, they probe, and wherever is the cancerous cells are there, especially they probe that one, tag that one, so that it will be killing only those cancerous cells. This is very interesting and very important. We also have to do this research in the lab. So same thing, photodynamic therapy. You can also see that chlorophyll. This chlorophyll, so it produces ROS, and in the cell, it kills all these. Apoptosis can happen. Then further cell death can happen. Through that, you can get a lot of the medicinal use or medical use or. This is the same thing, the photodynamic therapy. You can see that, wavelength, they use specific wavelength because the pigments have chlorophyll A, chlorophyll B, chlorophyll C, or, carotenoids, several other, pigments, bacterial chlorophyll, they have different wavelength it absorbs, so that you can use various light to kill the cancerous cells. So even say that body by kidney 24 to 70 hours. So, but it remains in cancer cell for a longer period of time when the photosensitizer cancer cell exposed to specific wavelength and produces abundance of oxygen that builds and kills the cancerous cells. So this is what now in the practice, and these are the apart from your food and other things, the therapeutic importance is also there. Okay, so now, so this is about the application part. There are people are now working a lot on the applications to understand and characterize and go for industrial application through photosynthesis. So, and I want to ask one question. If let's take, maybe I don't want to ask question, but I will tell you, if you increase photosynthesis, there will be a lot of other problems. What we do there, natural strains. Always we take natural strains for such kind of one. But you can also manipulate inside. Let's say I want to have lipids more, you can do it. I want to have pigments cannot be more. You cannot produce pigments because if you take pigments more, it may have others it can lead. It can synthesize. If you change something, pigments can be changed, but pigments itself cannot be improved in our research. But let's say there also like a Rubisco, for example. You cannot play with the Rubisco. If you disturb Rubisco, the entire system will collapse. So Rubisco, if you express, because it is it can produce a lot of other reactive oxygen species and can kill the cells also. Anyway, so now the just want to these figures might have showed already by many people. So now the pigments, since the topic is the pigments, where the pigment stays here. So this is photosystem 2 and a cytochrome B6F complex, photosystem 1 and ATP synthase. And when you have a green here, these are light arresting complexes. All together, photosystem 1 and photosystem 2. This is about the photosystem 1 have more pigment than photosystem 2. And this about 200 plus pigments will be there. 200 plus means 220, depending on species, about 218 to 20 is there. Going to this one, this have about 240 to 50 chlorophylls of photosystem 1. These two will have a pigments, high abundance of chlorophyll A, chlorophyll B and carotenoids. So the chlorophylls will be mostly present in light arresting complexes as well the reaction center. The reaction centers, I think they have explained, right? HRO has explained. These two will have the pigments, so which are very important to harvest the light. It can be chlorophyll A is the ultimate, they have to harvest and whatever the splitting process happens. So now why pigments are required again in photosystem 1? Can any of you tell why the pigments are required again in photosystem 1? Anyway, this is a linear electron transport, it goes. Why they have to have again pigments here? Anyway, so it's like a, if you like, if you just, then I don't have chuck this here. If you throw like this, it goes whatever, the from steady state to the excited state. So then it has to fall down. So what happens here, when it through all this linear electron transport up to here and comes here, and all this electron transport, proton motive force, they lose energy, right? When you are running marathon, have you any of you been marathon? How many of you there marathon? 1, 2, 3, only 4, 5. Yeah. So let's say 10 kilometer, 10K or 40K. 40K have you been anyone? 10K? Okay, 10K means 10 kilometers. So when you are walking or running, you will be exhausted. So when you exhaust, what they do? The guys will be there, they will feed you some juice or some energy drinks. So now the same thing, that means you are tired, you are losing energy. Similarly, you are losing energy here. By the time it comes to the before photosystem, I am tired, I want something. So what it does, you have to have energy again, solar energy, to pump these electrons to the linear transport. So that's why the pigments again mimicking to this one, but you don't need that much energy. The one you have ate in, let's say, a morning, it produce more, but in between you need a small, you need to have a little energy. That's why they have a longer pigmentation. They pick up, then transfer the energy to reduce NADP to NADPH, NADP to NADH. So, and here also core proteins and lighter, both have pigments, but light arresting complex have more pigments. And there are different kind of, there are a lot of things you can discuss. Each pigment have a different kind of, the energy, how they transfer, because so many pigments, it's like a network, how they'll be connected, dotted, they finally enter into the reaction center and we get. Okay, this is beautiful. I always tell my students, you have phycobilisomes, mimicking same photosystem thylakoid complexes. This is a thylakoid membrane, and sitting on the phycobilisomes, phycocyanin, phycoerythrin, allophycocyanin. These three pigments, different colors, and absorb the light and transfer to the photosystems. The rest will be almost same. So very interesting here. This is like a light arresting complex. Replaced, I know, in the higher plants they have a clear light arresting complexes. They are subunits, but those light arresting complexes are replaced by phycobilisomes in cyanobacteria. And the reaction center they have a green, again pigments. And interestingly, there won't be light arresting complex in cyanobacteria because it stays in the water. They don't require that much of energy. So whatever the energy coming from reaction center that have pigments, that solar energy is enough to transfer this one to linear electron transport. So these lakes are beautiful because of the cyanobacteria. This is a reflection where a Niagara or wherever you see the some beautiful when it sun shines and the pigments will be there. That means cyanobacteria or red algae or something, and they will be looking like that. Wherever you see that beautiful because of the algae or cyanobacteria. And bacterial chlorophylls having abundance of chlorophylls, rings like structures. They also have observe longer wavelength and then transfer the energy, the Q cycle, which is more of make into cyclic electron transport. So plants and algae and cyanobacteria, they have a cyclic and non-cyclic, but in bacterial system, they have only cyclic electron transport. I think Professor Raghavendra has discussed, where they will have they don't require that much energy. They don't produce NADPH. So it is only ATP it produces. That's why, it have, even in cyclic electron transport in plants, they have only ATP. Through cyclic electron transport, you get only ATP. That energy is enough to survive all the bacterial photosynthesis. It's mostly in anaerobic photosynthesis, this system will be happening. Okay, I think I will be at the end. This I think I'm not going to discuss because it's already HRO is discussing all this one. He is going to discuss all. So thank you, and you have any interactions? Not a question hours, but interaction. If you have, we can have any doubts, we can clear.
So in the photodynamic the therapy for cancer, usually ROS can diffuse out of these cancer cells as well. So it can go to the normal cells as well. So how this limit?
That's what I said. They have to, now the technology has developed. So you have probe. The cancer cells should be tagged with the pigment, and through that you are targeting directly to the cancer cells.
Yeah, after that the cell death will happen.
It may be there, yeah. Always side effects will be there, but it's more efficient. If it is probe with let's say even if you're giving chemo or something, the bacterial chlorophyll will be tagged, and this whatever the location you are in, let's say a tumor is there, you target only tumor. Yeah, cell diffusion will be happening, but the other cells can be come back again, right? They can grow again, even if it is that some side effects are there. But the cancer is always side effect. But the mechanism is the one, the ROS generation that kills the cancerous cells. But what mostly they probing, they tag, not probing, tagging with probing exactly to the inside the cell. So that's what the therapy. So you don't have questions. At least you can start your company. You see, like a lot of pigments can have people are now there earlier. Cinecostates was Pyrrolene now was the small scale industry. People are growing at home, backyard, and that there are a lot of in the villages, many of them still now do all these clothes or whatever, then they collect to industry people. Similarly, spirulina was backyard. The people a pond, now they grow just a simple water. You grow it and filter it, and you dry the powder, and the company people will come and collect the pigments. That was been there for now, because now the industry itself, they started their companies, and they're now producing more and more. So with the small scale, it's not, but still people are doing, specifically Mysore region. So there are a lot of people are now aware. They're in the backyard, they're growing and getting like a mushroom culture. You can do it at home. This algal culture also can be, you can grow, and you have to standardize. They will go and, if you go to CFTRA, have you heard CFTRA Institute? Food Institute, CFTRA in Mysore. So they will also give training on this algal culture, how to grow and what kind of conditions. Then you can implement at your home and get some money. I mean, precautions, there will be contamination can happen, other bacteria can grow, that we have to be careful. But, before harvesting, you have to check. Sometimes, toxic bacteria can grow, but we have to be very careful during that time. But, before, it's going to, the feed or whatever. And also, I forgot to tell you, this pigment, all this one also used for animal feed and biofertilizers. Nowadays, you can, algal culture can be, the waste of algal culture also used for biofertilizers. Yeah, there will be contamination that we should be careful. But in time to time, you have to see before harvesting, you have to see in the microscope. And if it is apart from your spirulina or something, you have to throw or see that what is the condition. But most of the algals, they are safe. It's edible. They're all edible. NADPH, bacterial photosynthesis, anaerobic photosynthesis, because there is no CO2, right? So, yeah. No, this will be used for other, the production of, all these molecules will be there. The ATP will be utilized again on the different cycle, not Kelvin-Benson cycle. So it will have to be, synthesized. Carbohydrate will synthesize, proteins, lipids, all of them will be synthesized. And most of the, the bacterial, they will be using this one as a bioremediation, all these bacteria. Okay, it looks like no interaction, but you might have understood everything, or maybe not. It's simple, I have not complicated story I didn't tell. It's more reachable and can be understandable. All of you can understand. And I said, one of the day, you can, all of you, that 15, 16 students, so will visit the industry by upstate. We have the, whatever, you're having food, that all industry, all, like, even there is one company working on leaf protein, Rubisco protein. So they leave from waste leaves, they isolate Rubisco, and Rubisco is again, rich of protein, abundance of, highly, abundant protein, where it can be used for a supplement of, or you can say vegan protein, veg protein, where protein replacing meat. They are working on those aspects. You can visit. There is some plant tissue culture related to plant, but many other, there are a lot of enzyme and biosimilars. You can go and visit the company. I will arrange this Saturday, or depending on who is our Jerome is there. What is the program? Can you see program? It will take time. But anyway, one of the day you will be going there. Okay, then if there are no questions, anyway, now I'll be continuing other two talks. You can also discuss any question if you have any question. Thank you.